So we downloaded the gene expression data from the GTeX website, and it comes in the form of a matrix with tissue samples and then the expression levels of each gene in the tissue sample.
The first part we did was a softmax regression model. So this is the softmax model. X, the input, is the vector of gene expression values for a particular sample. B is the bias, and W is a matrix of weights for the different tissues for each gene. Softmax is a function that turns this linear function into probabilities, and then Y would be the resultant vector of probabilities for each tissue type. W and B were learned using TensorFlow, using stochastic gradient descents over 2,000 steps. And each step, the input was a random sample of 50 samples from each tissue type. The resultant accuracy of the model was 0.93, and this was on test data, and a similar result was obtained on the training data. Once the softmax model was trained, we can use the weights matrix to select features. So the weight matrix has a weight for each gene for each tissue, and the genes that are highly weighted for particular tissues are discriminatory for those tissues. Each tissue, we took the genes with the highest weight and found their go terms online to validate whether they made any sense or if there were any interesting features there. And we found that most of the go terms made a lot of sense for the particular tissues that they were found to be discriminating for.
Using the gene expression matrix, we ran Pearson on all samples to create an all sample graph. On this graph, we ran spectral thresholding, which gives us a threshold value at which we can filter all other graphs through. We then took submatrices from the all samples matrix and running a Pearson correlation on each submatrix specific to tissue, we created tissue specific graphs. And we filtered each tissue specific graph to a thresholding value found through the spectral thresholding method. On each tissue specific graph, we ran paraclique, which extracts highly dense subgraphs of tightly connected genes. And then from these paracliques, we were able to run gene ontology enrichment to find possible gene modules.
So the results that we have so far are very preliminary, but it's very exciting how we could validate them against known annotations. And we have ideas to further integrate the two different methods we use and develop further methods to apply to this data.